In this video, I'm going to show you the structure of the GLUT4 transporter. In the previous video, you have seen that the GLUT4 receptor is present inside the cell in a storage vesicle. When insulin stimulates the cell, signaling pathway takes place and the GLUT4 receptor is moved to the surface of the cell. So the actual structure of the GLUT receptor is like this. The GLUT receptor is made up of 12 alpha helices, and these all of the 12 alpha helices they span the they span the plasma membrane. So we can say that it is an integral membrane protein, a transmembrane protein. So you can see the 12 alpha helices in this diagram. The amino terminal and the carboxy terminal of the receptor are present inside the cell. In the intracellular are present they are present on the intracellular portion of the cell. There is a loop, as you can see in this diagram, which is called as loop 1, and this loop has a glycosylation site and it is present in the extracellular space. There are other loops as well. The loop 6 is the largest one. Loops are the structures that are made up that join different secondary structures together. So in this case, the alpha helices are joined by amino acid sequences, they form loops. Between the transmembrane 7 and the 8, there is a loop which is called as loop 7, and it has a motif called STSIF motif. This motif serves as a binding site for the glucose. The actual receptor inside the cell and in the plasma membrane of the cell is not in this conformation, but in this conformation. So the 12 alpha helices they divide into group of two, and each forms a functional domain. So there are two functional domains that are present in this translocator. Six alpha helices they make one functional domain, and the other six make the other functional domain. So this is the 3D structure of this receptor, and I have taken this diagram from the PD. So this is the mechanistic diagram of the translocator. As you can see, there is a portion outside the cell. From outside, there is the glucose. There is glucose out and on the outside of the cell. When glucose is going to bind the receptor opens up at the extracellular portion. It widens and it exposes the pore through which the glucose can enter into the cell. The glucose then binds to the cell, and it starts moving towards the cytoplasm. So when the glucose binds and moves into the translocator changes its conformation. The outside portion gets narrow and the receptor closes at the outside, but it opens towards the inside. So the glucose can transport through the channel that is present between the two functional domains, and it moves through that channel and it enters into the cell. Now let's move towards a 3D structure of this receptor or translocator. In this 3D structure that is present on the PDB website, you can see the receptor. There are two functional domains that are shaded with different shades of blue. As you can see that this is the outside of the cell. This portion is the outside, the upper one, and the lower portion is the inside of the cell. Now the receptor is open at the upper portion, which means that it is open outwards. So in this conformation, the glucose can bind to this receptor. So in this conformation, the glucose can bind to the receptor, and in this conformation, when the outside of the cell or outside of the translocator closes and the inside opens up, the glucose can now enter into the cell. In this conformation, the glucose binds and enter into the pore of this receptor, and in this conformation it moves through that pore and it enters into the cytoplasm. So you can explore yourself on the PDB. I will share the link in the description of this video. Thanks for watching. like, share, and subscribe.